Okay, this next talk is structured as one of those keynote presentations where you feel like somebody's holding a fire hose in your mouth. And I'd be delighted if you interrupted, and I'm not hung up about finishing all of the slides. Okay? So interrupt me and be comfortable doing that. This is your one big dose of genetics this week. And it's aimed at the issue of individualized medicine. So I'm going to start by reminding you what the historical context is, and then tell you a bit about the kinds of genetic markers that are used. I want to show you, I think I've cut out my section on traditional genetic analysis. I'm going to go into a little bit on genome-wide association studies, which have become so important recently. I'm going to explain that their promise has not been fulfilled and try to help us understand why. Then I'm going to talk about genetic variation in drug response, which is mostly about cytochrome P450s, which are called CYPs, and about N-acetyltransferases, which are called NATs. And a little bit about genetic variation for susceptibility to disease, and hopefully then I will finish in time to give us a few minutes to all discuss what to do about individualized medicine and what to do about the day when patients walk into your office with their complete genome on a chip. And I looked it up on the web last week, and the current cost of the human exome is $1,688. So within a year or two, we're probably going to be living in a society where you can get the entire exome of a patient for $100. Which is down in the level of blood chemistry, okay, in terms of cost.
Question?
Yep.
Could you define what you mean by exome?
Yes. The exome is that part of your genome which is transcribed and which isn't, well, it's not spliced out during transcription. So basically that's the genes. And there's some stuff that gets spliced out that's important. Some of the microRNAs and things like that are spliced out. But the structure of eukaryotic gene is introns and exons. The introns are spliced out. The exons are spliced together. They code for the proteins. The introns get recycled. And the amount of DNA that you have to sequence to get the exome is only about, I think, 1% to 3% of your genome, which is why it's cheap. Sequencing the entire human genome involves sequencing an awful lot of DNA, which is not being transcribed and making proteins. So the exome is the part that is transcribed and makes proteins.
Right, so it would be a cDNA library then? Those things that are actually
Yeah.
produced in RNA, and then what tissue would this exome be from? Because what's expressed in the liver is different than what's expressed in the lung.
My guess is that my guess is that it's either from a blood sample or more, or it's probably either from a white blood cell or it's from a tissue swab in the cheek. Okay.
But those would only be the genes that were expressed in those tissues then, right?
It's genomic DNA.
Genomic DNA, where bioinformatically you define these are the sequences that we need to know.
It's not the transcriptome, okay? It is that part of the DNA which is in exons. Okay? I don't do it myself. If somebody here does, please inform us. Okay, I just want to start by reminding us of our ancestry, because this is where our genetic variation came from. This is about the last 7 million years. So we start off with Sahelanthropus. We go up through a bunch of Australopithecines. Then we have some Paranthropus. And then we get into the Homo lineage. We started using tools about 3 million years ago. Fire, which is very important for our diet and for cooking and for lung cancer, probably started about 2 million years ago. We probably started having birth problems with a narrow birth canal about 2 million years ago. It looks like cooperative hunting in a sort of Homo scale came in up here, although I must say that chimpanzees were cooperatively hunting down here. And everybody who came out of Africa about 100,000 years ago has got some genes in them that resulted from hybridization with Neanderthals. So we all have a bit of Neanderthal gene in us. By the way, that was made possible by the recovery of fossil DNA from Neanderthal skeletons and the assembly of the Neanderthal genome just in the last couple of years by Svante Pääbo and his group. So when we look back on issues of medical interest, we've been bipedal for about 7 million years. We've gone through a sequence of different kinds of diets. Australopithecines were probably frugivorous. There were probably other things that they ate. Interestingly, when we think about the biodiversity on the planet, there have been times when there have been several species of Paranthropus and Homo that coexisted. So we kind of hope that they're out there. People keep dreaming of Yetis and Sasquatch and things like that. They're not there, but at times in the past, things like that were there. Homo erectus came out of Africa and colonized Asia and Europe well before Homo sapiens. They had fire, cooking, hunting, scavenging. There were hand axes in Spain 800,000 years ago. In fact, near Rioja, in a cut for a railroad line, they uncovered a cave that indicated massive scale cannibalism. There is a cave full of bones 5 or 6 feet deep over, I understand, about an acre or something like that, every one of which human bones, and every one of them has got a hand axe scar on it. So they were processed for eating. I'll show you this in more detail, but I'll repeat it several times. We came out of Africa about 100,000 years ago. We paused in the Middle East for 30 to 50,000 years, and then we spread out into Asia and Europe, and those that did that hybridized with Homo neanderthalensis. By the way, I think this is more titillating than significant in terms of the portion of our genome. We probably have more virus genes in our genome than Neanderthal genes in our genome. So just a couple of things for numerology. These two statements are both true. So I am 99.9% like each of you, and each of our genomes can still differ at up to 3.3 million positions. That's kind of the basic thing that you need to know if you're going to get into personalized medicine. Okay, that's a very, and this is also essential, as I think we'll see as we start to discuss whether the different group ethnicities on the planet differ genetically. A little bit about some of the markers that are used when geneticists look into the genome. For, 15 years ago, they were using microsatellites. These are short tandem repeats, so you can pick those up as bands when if you chop up DNA and put it in an ultracentrifuge. These are repeats of 2 to 6 nucleotides. They're interesting in medical genetics because they're involved in things like Huntington's disease, Fragile X syndrome, trinucleotide repeat disorders, spinocerebellar ataxias, myotonic dystrophy, things like that. In my own life, it's kind of tragic that my co-author on my evolution text, Rolf Hoekstra, who had been thinking about what his father died of, decided to have himself sequenced to see if he had a trinucleotide repeat disorder. He does. The number of repeats is predictive of the age of onset, and Ralph will start to have difficulty walking, chewing, breathing, all that kind of stuff when he is about 70. He's currently 66. He took early retirement and started sculpting. I know a number of cases like that where an evolutionary geneticist has a genetic disease. Copy number variants are something that if you haven't heard of before, you're going to be hearing a lot more about. And I have another couple of slides on them. These are DNA sequences that are pretty long, and their copy number varies among individuals. And they're increasingly viewed as being the kind of variation in normal human populations that has a lot to do with disease risk. Okay? And then the one which has become extremely popular since the sequencing of the human genome is the single nucleotide polymorphism. You can see one right here. Okay, so you'd have a long sequence in 2 people, that was exactly the same except that they differ by one position. So that's a single nucleotide polymorphism. The convention on this is that you will recognize it as a polymorphism when the less frequent allele in the population has a frequency above 1%. And there have been 28 million of these discovered, 6 million have been validated, 4 million are in genes. These numbers are going up. So this is just an estimate of, what portion of the genome is actually being transcribed and we can call a gene. Much smaller than the total number discovered. Okay, a little bit about copy number variation. This was the paper in Science that brought home just how significant this could be. It's more technically difficult to discover copy number variation than it is at this point to discover single nucleotide polymorphism, which is why the sample is a bit smaller than you would normally see in a SNP study. So they just looked at 20 individuals and they found 221 copy number differences representing 76 unique copy number polymorphisms in these 20 individuals. So on average, you and I differ by 11 different copy number polymorphisms, and that one of these things on average is about 465,000 bases long. That's a lot of variation. So in this one study with 20 people, they saw a copy number variation in 70 genes that were involved in all kinds of important biological functions, and some of them were associated with disease. And other studies, Bernie Crespi's work has shown that some copy number polymorphisms are indicative of, at pretty high level, autism and schizophrenia. Now let's go to SNPs, okay? And I'm going to spend a little time on this slide. I really love this study. I think this is a landmark study, and it will have follow-ups that will go into greater detail, and we'll learn more. But this was the first time that people had really looked at human genetic variation across the planet. So this is human genetic variation. They took 938 individuals. They came from Africa, the Mideast, Europe, Central South Asia, East Asia, Oceania, and the Americas. So here they're dealing with Native Americans. So the 938 people are lined up along this axis here, and the 650,000 single nucleotide polymorphisms are aligned on the y-axis, okay? And you can immediately see a couple of interesting things. Africa is relatively homogeneous. The Middle East is mixed. Europe is fairly homogeneous. Central South Asia is fairly homogeneous. East Asia is fairly homogeneous. Oceania is fairly homogeneous, and the Americas are fairly homogeneous. So that's 1, 2, 3, 4, 5, 6, 7 groups. Remember that's 7. It's going to turn up in a slide later. Now, they then asked, okay, what can we say about our evolutionary history if we use that variation to look at a phylogeny? Well, what they recover basically is, yes, if we just look at the genes, we don't know anything about any other information, fossils, archaeology, history. The genes say we came out of Africa, and they say we probably came out about 100,000 years ago. The length of this segment here indicates that we probably went through a genetic bottleneck when we came out. Most human genetic variation is in Africa, and the rest of the world actually has a small portion of it because we actually spent most of our time evolving in Africa, went through a bottleneck, and then spread out through the world. Then we have people in the Mideast indicating that's where we paused. Then the Europeans went into Europe. People went into South Asia here, went into East Asia here. We have a few estimates of when that might have been based on other kinds of evidence. We think that the Native Americans probably came over the Bering Strait at about 14,000, and that the move into Papua New Guinea and into Australia was at about 40 or even 50,000 years ago. Now, from the point of view of personalized medicine and some of the stuff you're going to see later on drug metabolism, the point is that we have, as we spread across the planet, differentiated into different lineages that encounter different diets, different diseases, and different physical environments. And we not only have differentiated genetically in response to selection caused by those factors, we have differentiated just simply because of sampling and molecular genetic drift across all of that process. Now, of course, this is the kind of picture that only could have been made at a point in history where we could still find people who were plausibly of these ethnicities and these historical backgrounds. And intermarriage and global travel would make a study like this extremely difficult and probably impossible, maybe within another 1,000 or 2,000 years. So it's kind of a fortunate accident of history that our technology and our ability to get this information coincided with a point at which the samples still existed. Okay, now just a couple of little other things that happened with this data. Two things over here. This, the green here is the within population variation. The blue is among 7 group, okay? And then this is among population within group. The red is among population within group. This is the result of analyses that basically say this is how much variation out there is due to within population. This is how much, this is just for the X chromosome. And then this is for microsatellites in the genome. Okay, so this is autosomes. This is one of the sex chromosomes. The important point here is that almost all of the genetic variation in human populations is among individuals, and it's not accounted for by which ethnicity. You can see that at no point is one of those, or is that classification by 7 groups accounting for more than about 12%. And in fact, if you look across the largest part of the genome here, it's accounting for about 9%. And the particular cluster within that, the population, is only accounting for 2 or 3%. The other thing that's kind of interesting is that if you center the process on Addis Ababa, so you say, well, we came out of Africa and we went across the globe, this is the heterozygosity of the populations, and this is what happens in Tierra del Fuego. Okay, so this is where the process ends in southern South America, and you're losing genetic variation all along the route. So the highest genetic variation is up there in Africa, and you're losing it as you go out along that tree. Now, the first important, well, let me go back just a sec. These are principal components, which are summaries across all of those 650,000 SNPs of the principal axes of variation. And they allow you to break out, for example, in Europe or over here in the Middle East, some groups. You can see clusters at that scale. And you can say, oh, there really are genetic differences between Russians or the Basque or the French or the Druze or the Bedouins or things like that. Well, this is how much variation is being accounted for by those differences. 2% on this axis, 2.4 and 1%, 1.6 on that axis. So if you go down to the real fine scale, because you've got so many SNPs and you've got such a large sample size, you can dissect out some genetic differences and say, yeah, there are some tendencies for people to be different from each other, but if you add those two up, let's see, that's 4%, they're 96% the same. Okay? So this is pretty nuanced differences. Yeah.
It's amazing how it follows the geographical distribution.
Well, yes, but it makes sense that people who are close to each other in space breed with each other. And actually, Steve Jones has got a nice note that up until 1830, with the invention of the bicycle, the average distance between partners in a marriage between their birth was about 1 mile. And that with the bicycle, it jumped up to about 15 to 30 miles. And now with the jet airplane, thousands of miles is not unusual. Okay, so this is a relic of the time when people walked to find their partners. We don't know that. I'm totally agnostic. My guess is it's a mix, and from what I know just in general about the whole argument about a selection and drift, is it's going to be hard to see, hard to tell, and a lot of molecular geneticists, evolutionary geneticists have concluded that much of the time, actually asking whether it's selection or drift doesn't help very much because our power to discriminate those two hypotheses is not great. For particular genes with particular histories, in one particular population, you may be able to pick up a strong signal of selection. The lactose stuff, for example, is one of those. I'll show you a few cases later where we have information on whether selection is operated on disease resistance genes. And what's most striking about it is how few diseases and how few genes really show positive, strong evidence. Okay. This is the summary slide on are there human races? The answer is that whole idea that ethnicity or skin color or something like that gives you very much explanatory power is not very great. These are studies that accumulate from 1972 with proteins up to the SNP study that I just showed you. This is probably the most reliable one. 88.9% of human variation is among individuals. 2.1% is among differences like, are they French or Russian, or are they Druze or Bedouin? 9% is among those 7 big geographical groups. Okay. This is the most important cultural message of evolutionary genetics of humans. Race is a social concept. It is not a biological concept. However, if we're 99% identical to chimps, 99.9% identical to each other, and if 85% of the remaining 1% of our genetic variation is among individuals and only 9 to 10% among ethnicities, then why can doctors continue to find ethnicity a helpful indicator of genetic risk for certain diseases? They do. Sure.
More likely it's a proxy for differential environments.
That's true. And in some cases, like the Ashkenazi, which have certain genetic diseases, or the Afrikaner, which have certain genetic diseases, the Ashkenazi may have it because of a history of exposure to particular diseases. The Afrikaner, because of a founder event. And we actually know the name of the woman, and we know in a marriage in 1630 going into South Africa that had the gene in her body that then led to the high prevalence of those diseases in South Africa. So that's another reason. I think the reason really was in that slide where I showed you that despite the fact that only a very small portion of the variation is accounted for by what we would call tribes or ethnicities or something like that, that we have sufficient genetic power to discriminate, that for particular genes, that can still make a difference. The fact that over the whole genome we're 99.999% similar doesn't mean that for one gene, it might very well be assorting very strongly in a particular population. Okay, so if you do a traditional human genetic analysis, the probability in a random sample of a disease is low, so candidate loci have been identified by a long history of sampling large populations. Those kinds of genes have high penetrance. That means that the probability of getting the disease when you've got it is high. When you have that genetic variant is high. The effect size is high, so the detectability of the disease is high. And that results in a human genetic disease like cystic fibrosis that has a very high odds ratio, which is the probability of being affected when an allele is present relative to being affected when it's absent. So that kind of traditional analysis uncovered genetic causation of rare syndromes caused by single genes with large effects. Here's an example. This is hemochromatosis. So this is the commonest recessive deleterious genetic disease in Europe. It's at a frequency of about 8 to 12%. So 1 in 300 to 1 in 600 people in Europe is a homozygote. It's rare in Asia. Middle-aged men get this disease, okay? They get it because they don't menstruate. They have increased iron absorption, and they can have liver fibrosis, cirrhosis, liver cancer, cardiomyopathy, diabetes, arthropathy, hypogonadism. Males don't like hypogonadism. And they get dark skin. Interestingly, when I was interviewed about this disease once on PBS, the guy in the broadcast booth who was the technician had it and came out and showed me how dark his skin was. Okay, it was dramatic. So it's a single mutation. And here, note that it's rare in Asia. The selective disadvantage is small because it's a late life onset. It's middle-aged men. They've already had their kids. And here are two hypotheses. One is sort of the classical human genetic response to a human genetic disease at high frequency. Oh, it actually had a benefit in the past. We're seeing something like sickle cell anemia, and that's what drove it to high frequency, because the model that most people have in their minds is sickle cell anemia. And the number of cases of genes that actually function like sickle cell is extremely small. Okay? But that would naturally occur to somebody. Oh, there was plague or TB or typhoid in the past, and it confers resistance to that, and therefore, that's why we still have it at high frequency. It's going away because we don't have those. Alternative hypothesis, it's a benefit to women because they lose iron in menstruation, and everybody has a mother and a dad, and so the dads got it from their moms, and it's not causing them too much problem, okay? But it's enough of a benefit in women to keep it around even though it's a problem in men. That's hypothesis 2. So do you think those ideas can be tested? Do you think the frequency in Europeans, 8 to 12%, is a mystery calling for explanation? And do you think that if it protects against plague, then how do you explain why it's so rare in Asia? And if you think that it benefits women, why is it so rare in Asia? Those are problems. It's not clear why hemochromatosis is at high frequency in Europe. And there are some really puzzling things about its geographic distribution. It may be that these are both right, and the mutation that caused it originated in the European lineage and simply wasn't available in Asia. But it's at low frequency in Asia. It's not absent. So maybe it just got there late. But you can see that this is a classical scientific situation. We have alternative hypotheses. We have a mix of kind of conflicting data. None of that, about which, frankly, I think people should remain agnostic. If you don't know why something is there, you should lay out the alternatives and then say, but we can't decide, which is what I'm trying to say to you right now. That has not prevented doctors from writing popular books claiming that it was caused by plague. Okay? Yep.
Just as the sickle cell trait is found in the malaria belts, isn't it possible that there were iron-sensitive bugs that were localized to Europe and not seen in Asia?
There were iron-sensitive what?
Pathogens that were more localized to Europe than they were in Asia.
Right, but plague certainly was not one of them, was it?
But there may be many iron-sensitive killers out there, right?
There could be a lot of iron-sensitive killers, but then you have to argue that, you know, all bacteria need iron. So you have to argue that bacterial disease was a more prevalent cause of mortality in Europe than it was in Asia. And that just seems to be implausible on the face of it. But maybe it may be so. Okay. Now let's take a look at genome-wide association studies. These are a product of the sequencing of the human genome. And the sequencing of the human genome, which cost a tremendous amount of money, hundreds of millions of dollars, maybe billions at the time, was largely based on the potential health payoff. And the idea was you could use SNPs and sequence information to look for common chronic late onset diseases where you have low relative risk, where you've got a number of genes, polygenes, that are affecting the disease risk. They can work when these variants, these alleles, are present in many individuals in the sample with a penetrance as low as 10% and a relative risk as low as 1.1. 1.1 times more likely to get the disease with it than without it. And that led to the HapMap project, the haplotype mapping project. Find markers all over the genome and associate them with diseases. One of the early landmark studies came out 4 years ago. This is a fast-moving area. This is the Wellcome Trust Case Control Consortium paper in Nature. They looked at 14,000 cases of 7 common diseases with 3,000 shared controls. They broke that down in many different ways. They were using Affymetrix chipsets. They had 2,000 individuals examined for 7 diseases compared to 3,000 controls. So that's where the 14,000 comes from, 7 times 2,000. They picked up out of all of these, by the way, that should be the 500,000 Affy chipset. Out of all of those SNPs, they picked up 24 association signals. You have to have a very, very high statistical significance because you're looking at so many tests across the genome. The probability that you could get a significant association at random is pretty high when you do so many repeated tests. So you normally see in genome-wide association studies, instead of p equals 0.05 or 0.01, you ask for a p equals 1 in 10 million, okay? Because you're doing so many repeated tests across so many genes. What they picked up was, oh, we have a gene for bipolar disorder, or we have a SNP associated with some gene for bipolar, 1 for coronary heart. They picked up 9 for Crohn's, 3 for rheumatoid arthritis, 7 for type 1 diabetes, and 3 for type 2 diabetes. Some of these are conferring risk for more than one disease. There's a bit of overlap in the diabetes hits. And most of these loci are having pretty modest effects, okay? So they emphasize the need for large samples. We'll come back to that. This is what it looks like. Here, for example, are the hits for rheumatoid arthritis and type 1 diabetes. This is a Manhattan plot. So basically what you're doing here is you're starting with chromosome 1, and you're putting out the whole human genome out through chromosome 22 and then the X chromosome. You're seeing 500,000 SNPs examined across the whole genome like this, and they pick up a really nice, highly significant hit here. This, by the way, this axis is like 10 to the minus 15th probability, 10 to the minus 10th probability. You want to get above about 10 to the minus 7th. That's highly significant. Sitting right in the middle of MHC, you have got a signal of genetic variation in the sample that is indicative of susceptibility to rheumatoid arthritis and type 1 diabetes. There's a really nice hit on chromosome 9 for coronary artery disease. Those are the significant hits for Crohn's disease and so forth. Okay, this was the first really big, really well done case control study using genome-wide association. However, this promise is not really working out very well. And it's not working because even though those are highly significant hits, they're actually only explaining 2 to 3% of the variation in the disease. There's a big difference between statistical significance and amount of variation explained. You can have a highly significant, very small effect. And that is characteristic of genome-wide association studies. Okay? Now, if we look, at what we know about human genetic disease, we've got nearly 3,000 disorders that can be traced to specific genetic defects. Most of those were done by classical methods. We've got another 3,700 that have some evidence of Mendelian inheritance. If you want to, you'll get this in a PDF. you can go to that site and find all this information. That's up on the web. Here are some reasons. These common alleles, the things that are picked up in the genome-wide association studies, almost never have a very large effect, and they explain little of the variance in disease. 1% to 2% is normal. The unexplained genetic causation might be due to low penetrance alleles. It may be missed because it's epistatic or it's caused by rare genes of large effect. Epistatic means that it takes 2 or more variants interacting with each other to elicit the phenotype, okay? And that's not picked up in a normal genome-wide association study. The rare genes of large effect are not really on the chip at all because their frequency in the population is below 1%, and so they're screened out when they build the chip. So the technology screens those out. And by the way, the argument is that the way to get at these is to sequence the whole genome, and now that you can get an exome for, $1,500, $1,600, that day is coming. We may pick up some of that when we look at rare genes of large effect. And it's certainly true that the genetic effects are masked by environmental variation. Most of these diseases are, all diseases in some sense are genotyped by environment interactions. And especially things like diabetes and heart disease have large environmental effects. So to summarize that, the haplotype map, that means the map of sections of the genome that are marked by these SNPs and that are moving together under recombination. That's a chunk of genome that we can call a haplotype. That was made possible by this, but we've really not identified very many alleles that have significant impacts on common diseases. Effect sizes are small, causal links to the diseases are obscure, and I think we have to say at this point, the promise of individual medicine based on gene chips has not yet been realized. There's a lot of discussion of it, but if you actually get down to the practicality of it and you try to say, how confident can I be that a person who has this genome rather than that genome is at this risk rather than that risk, well, we're not very good at it. And so when we look back at some of the rhetoric that justified the huge expenditure to sequence the human genome, I think we have to realize that the benefits that we're getting from that are not the direct benefits in individualized medicine, at least not yet, that had been expected. But we are picking up, of course, a lot of other indirect benefits. I'm not saying it was the wrong thing to do. I am just kind of amused at the way that rhetoric and sales jobs have a huge technological impact on science. Okay. So Alan, I'm now going to talk a little bit about genetic variation for drugs and pathogens. Yeah, Steve.
Can I just ask about the environmental masking? Do these studies, for example, adjust for confounders like obesity as a proxy for environmental effect? Is that a way to get past the environmental masking as a problem?
Yeah, some of that's been done. Sure, you can build a linear model in which you make your genome-wide association study more complex, and you build in adjustments for obesity. You can build in adjustments for anything that's been reliably measured. The limit there, though, is that the genetic variation is always much more precisely and exhaustively measured than the phenotypic variation. It's much harder to get phenotypic variation. It's expensive. But yes, where you've got it, it's been used. Okay, now a bit about human genetic variation for responding to drugs and pathogens. And I'm going to talk a bit about the major effects. I'm going to talk a bit about interactions with sex, age, ethnicity, and geography, which all are pointing towards things like personalized medicine. And the evolutionary questions in the background are, why does this kind of human genetic variation exist? Why is it maintained in populations? And what consequences does it have for health and disease? So, this gets us into something which is called pharmacogenomics. And that means the impact of individual genetic variation on ability to metabolize drugs. This field actually got founded in the 1950s in a notorious tragedy that happened after Ciba-Geigy had tested cleoquinol on Europeans and had found it safe, and it released it in Japan without testing it on people of Asian genetic ancestry. Okay? It killed, I believe it killed somewhere between 12 and 70 people. I forget the numbers exactly, and hundreds were seriously compromised. They didn't die. After that, the fact that cytochrome P450s, which are henceforth going to be called CYPs, the variation among individuals and among ethnicities in cytochrome P450s and in N-acetyltransferases, also called NATs, was also found through adverse drug reactions. So drugs have been developed and released, and or either were not yet fully tested or were released before being adequately tested, and some people had some really bad reactions. Interestingly, and I think this is quite important because it links to cancer, the same gene products that metabolize drugs, the CYP and NATs, are involved in processing toxins in food and smoke, and thus mediate the mechanisms that elicit cancer and that shape the evolution of the resistance of cancer to drugs. So these are the kinds of things that are involved in the mechanisms of resistance to chemotherapy, but they are also involved in the genotype by environment interactions between environmental factors and cancer risk. So it's not just drugs that we're talking about. We're talking about things that have multiple functions and that are important ways in which our bodies deal with toxins. So a little bit about the cytochrome P450s. They're an amazing family. Okay, they all have a heme group, and it's actually the color of that you get out of these heme protein complexes that led to the name chrome for chromatic or color. And just a general biochemical comment, anytime life has really wanted to manipulate very precisely oxidation reactions, it often uses a heme group. Okay, so these are involved in oxidative reductive reactions. We have a family of 57 of them, and they bind to our endoplasmic reticulum. They're on the inner mitochondrial membrane, and they mediate steroid metabolism as well as toxin metabolism. In the first 3 families, there are 23 genes, and we're going to look at one which is called cytochrome P452D6, okay? So I'm now going to focus way in. You can see that there is a wilderness of these things out there. And believe me, drug companies know a lot about each one individually, and they are most worried about that one. At least one of the ones that they're very worried about. This is a gene whose variation is quite important in the clinic. It was picked up in 1977 in a study where a volunteer had a hypotensive response to a drug called dibrisequine, which is an antihypertensive. And in Germany, in another study, a volunteer had a side effect taking spartiene, which is an anti-arrhythmic. Now, this kind of variation, this particular gene, is known to be involved in adverse reactions to all of these other things, which you can see listed there, including tamoxifen. This is one of those cases where actually there is significant genetic variation among groups, among ethnic groups. Here is a table of ethnic groups. Here is a table of the different alleles that occur at this locus. You'll notice there aren't just 1 or 2. There are actually probably more than 17, but these are the common ones. And you can see that the Chinese have a lot of allele 10, and Americans have a lot of allele 1. And these process drugs at different rates. But I think if you go across that, you'll see that most populations have more than one allele, and that if you tried to base your drug prescription just on noticing that a person was American or Chinese, well, in some Chinese populations, you would be just flipping a coin because they're at 50% for that allele over there. In one American population over here, just over a quarter of them have it, and another subgroup of Americans has 90%. So just using that kind of crude descriptor isn't very good. You really know what does that individual actually have. And if you look across, this is variation in a number of different cytochrome P450s. This is the one we've just been looking at down here. This is an N-acetyl-transferase. And I think that if you just look at the percentage of, this is now microsatellites rather than particular alleles. Pardon me, this is a structure-defined subcluster giving us different populations. And what we have over here are different frequencies within those subclusters. You can see that none of this kind of ethnicity information is giving us 100% accuracy. Sometimes you get pretty close. You get to 90% accuracy on this one up here in, now I'm getting tied up in this cord. But I think the take-home message is that ethnic information is not a sufficiently accurate predictor to make a physician comfortable in deciding that he knows what version of a gene a person has. I mentioned copy number earlier. This particular one, 2D6, can vary from 2 to 13 copies for individuals. It metabolizes 25% of the drugs on the market. And the individuals taking it have between 2 and 13 copies of it, okay? Taking those drugs have between 2 and 13 copies. Its variation influences 12 to 14% of drug metabolisms, okay? So that's, of that 25% of drugs on the market, the variation at this locus is actually significant for about half. And that's antidepressants, antiarrhythmics, anti-cancer drugs. You know, you think about why if you go to the shrink and you ask for something for your depression and he has to cycle through a bunch of them and you might end up on Lexapro or something like that, well, it may have something to do with how many copies of this gene you have at this locus. The duplications in this gene are reducing efficiency of drugs that treat arrhythmia, and they actually have a huge effect on how likely it is that a person will become addicted to heroin or will respond to methadone treatment, but they improve tamoxifen treatment of breast cancer. So that's an important kind of a thing for an oncologist to know. Now, the drug industry has decided they want to avoid developing drugs because they can't predict how individuals react to this particular thing. So they're actually trying to work around this a bit. I suppose that if they could be confident that individualized medicine was available worldwide, they could be confident about releasing the drugs, but otherwise they are actually going to be running into some legal problems with serious side effects. The N-acetyl-transferases is a somewhat similar story. They activate and deactivate drugs and carcinogens in the cytosol of the liver. They're located in a different part of the cell. You could have rapid, intermediate, and slow acetylator phenotypes. Same kind of historical story. A patient was being treated for tuberculosis with isoniazid, and it had a mutation in an N-acetyl-transferase. They metabolize sulfonamides, metabolites of caffeine, many other drugs. There's a lot of detailed information on global variation here, but just to indicate it, Europeans and North Americans tend to be slow acetylators. East Asians are much less frequently slow acetylators. And the big point I want you to take home from this one is that we know something about the G by E interactions. So, if you are a slow acetylator for N-acetyltransferase 2, you're 1.46 times more likely to get bladder cancer. However, if you have that combination of that allele plus an NAT1 fast acetylator, and you ever smoked cigarettes, you're 2.73 times more likely to get bladder cancer. Okay, you see the interaction effect? You got to put the two different genes together and throw in an environmental thing, and then bango, you were starting to explain a fair amount of bladder cancer. Colorectal cancer. The fast acetylators have higher risk than the slow ones. Smoking increases risk. It does so among carriers of both the 1 and 2 fast acetylators. And that status is affecting the relationship between smoking and risk. Pancreatic cancer. N-acetyltransferase is interacting with dietary mutagens to increase risk of pancreatic cancer. And if you put in both the diet and the genetic variation, you get risk factors of 2 to 2.5 times risk ratios, but you only get it in men. You don't get it in women. So there's an effect of sex here. So you can start to see just how specific the genetic interaction is with both the environment. There's probably going to be some information coming in here from ethnicity, but if you actually know the genetic variation, you can forget the ethnicity and just go with what you've got from your gene sequence. And the interaction with the environment appears to always be important. So the take home here is that individuals vary in the genes, the gene products that process drugs. If they can't process it rapidly enough, it'll build up and can act as a poison. Cancer cells can also vary. Cancer clones can vary. Mutations can occur in cytochrome P450s and in N-acetyltransferases in cancer clones. And they therefore also vary in susceptibility to chemicals in the environment and to the drugs used in chemotherapy. Okay, a little bit at the end on what we know about human genetic variation for disease resistance. Okay, so basically the structure is, I've showed you that there's a lot of human genetic variation and historically where it came from. I've showed you that some of it is clinically important because of drug metabolism, and some of it is clinically important because of disease. This is data from Adrian Hill at Oxford and Arno Motulsky at the University of Washington in Seattle. And it shows a fairly up-to-date summary of our knowledge of what genes, what immune genes are known to be associated with disease resistance. And just let me count, I'll name the diseases: TB, AIDS, malaria, hepatitis, typhoid, leprosy. How many diseases are there? Thousands. Okay? Good data in the immune genes for just this number. Non-immune genes, how many? Okay? Malaria. I mean, malaria has obviously put a big signal of selection into the human genome. Leprosy, TB. This is a particular kind of vaccine, actually, and HIV. We have signals there for HIV. That's it. Those are the places where we have really good signals of selection in the human genome, that there is a gene for resistance that has had a history of exposure to epidemic disease. Okay? Not many such genes. I'll just talk briefly about the ABO polymorphism and then wrap up. So you all know what that is, and you know that it has a lot to do with blood transfusion and who can take blood from which type, and here's a table of recipients and donors. Okay, and just, I think it's culturally interesting that some people have two different ABO blood types because they shared blood with a non-identical twin before birth, and that's actually one of the ways we can diagnose how many people actually had a twin before birth but were singletons at birth. They're mosaic for ABO. This is what we know about ABO and disease resistant. A is more susceptible and O is more resistant to falciparum malaria. O is more susceptible, B is more resistant to Norwalk virus, which is an important diarrhea that can cause a lot of infant mortality. O and A are more susceptible to Helicobacter pylori, and it adheres to the gastric epithelium using antigens associated with O. Blood group O is more common in isolated populations. You see a lot of it as you go south in Native Americans from Central America down through South America. Susceptibility to syphilis, cholera, and plague depend on this polymorphism. And if you have a mutation that causes reduced secretion of these antigens into saliva, then if you're homozygous, non-secretor, you're at higher risk of infectious disease like meningitis, but secretors are at higher risk of upper respiratory tract infections like flu and all of the different colds and things like that. So you can see there's a trade-off there. So, comments on individual medicine. Individual genetic variation has important effects on treatment efficacy. Proper treatment, really precise treatment, would require determination of each individual's genetic state. Geographic evidence suggests the evolutionary process that produced this variation probably involved natural selection to local conditions, plus drift, I should say. And to summarize the lecture, this resulted from our spread across the planet, our genetic drift in doing so, and our exposure to different environments. There is widespread genetic variation for drug response, and it produces lots of variation in side effects and susceptibility both to treatment and to cancer, response to treatment and susceptibility to cancer. We have identified genetic variants for resistance to malaria, leprosy, HIV, typhoid, and TB. The patterns we now see could have been produced by a loss of resistance as well as by its acquisition. And proper treatment is going to require very precise determination of individual genomic state. So the company that is charging $16.88 is Otogenetics. You can look them up on the web. So, do you guys think that the benefits of individual genotyping would outweigh the costs? Does the answer that a person gives depend on whether they're healthy or sick, thinking about paying their insurance and the games that insurance companies are going to play with this, or thinking about their own state, the probability that they'll die? Should drug companies provide patients with information in their drug handouts on the potential range of individual responses? And why does so much genetic variation exist in humans for enzymes that metabolize environmental chemicals? Those, I think, are some of the big questions. There are many more. It is now 10 o'clock, and I'm going to ask Randy what you want to do. We have a couple of
Yes, so group photo right now, and then gift shop is open until the break is over. Then towards the end of the break, we'll do the evaluation.
Probably we'll talk about that in a moment. But for right now, unless you want to take one or two quick questions, Steve, in fact, I think we have. We don't really have.
We don't really have much time. I think I ought to probably take one question.